a bacteriophage? A virus that attacks a bacteria is called as a virus that attacks a bacteria is called as a right. So today we will talk about the bacteriophage and we will talk about how to treat viral diseases, right? Those are things we are going to be talking about. Fine. Do you know what is an inclusion body? Do you know what is an inclusion body? What is an inclusion body? If a virus or its proteins are deposited in a cell, if a virus or its proteins are deposited in get in. If a virus or its proteins are deposited within a cell, it produces, listening, audible. Let's say there is a virus. If this virus with its proteins deposits in a cell, it will be called as an inclusion body. Now, why is this type of an inclusion body necessary is, let's say I have been infected with cytomegalovirus. I am infected with cytomegalovirus. I need to know, right, like when I do a histopathology, I have to see if cytomegalovirus in the kidney, when you take a cross section, you will see cytopathic effects. That is detected through inclusion bodies. In cytomegalovirus, this is a kidney, let's say. This is a kidney. In cytomegalovirus, it causes owl's eye appearance. It causes owl's eye appearance. Owl's eye? So this is an inclusion body. This is also a cytopathic effect. This process of change within a cell. The cell was a normal cell. The cell earlier was a normal cell, correct? That got changed into a different type of cell now. These proteins are called as inclusion bodies. This change is called as cytopathic effect. It's called as cytopathic effect. Right? Understood? Okay. Now, if a respiratory syncytial virus comes, it causes degenerative change. It causes fine. If a herpes simplex virus comes, it causes ballooning effect. It causes ballooning effect. So there are various. So you understood what is happening. Now, normally a cell will be like this, with a nucleus, cytoplasm, mitochondria, right? But after a virus attacks, it will have a new inclusion body. Right? So that process is called as cytopathic effect. Right? Now I will give her down a list of cytopathic effects. Write it down. First, I'll tell you. So you understand what is inclusion bodies? I'll give you a small definition. Just write it down. The aggregates of virus or virus proteins. The aggregates of virus or virus proteins which changes the staining property. Is called as an inclusion body. Changes the staining effect. Staining property is called as a inclusion body. There are two types of inclusion body. Intracytoplasmic, intranuclear. Right? Okay. Write down. Nuclear, intracytoplasmic. Nuclear cytoplasm. Fine. Right. Write down examples. Negri bodies. Negri. N-E-G-R-I. Negri bodies. Rabies virus. Hyphen haki. Write down Negri bodies. Put a hyphen and then write rabies virus. Right? Write down Taurus bodies. Taurus. T-O-R-R-E-S. Taurus bodies. Yellow fever virus. Yellow fever virus. Write down molluscum bodies. Molluscum. M-O-L-L-U-S-C-U-M. Molluscum bodies. Molluscum virus. Molluscum virus. Molluscum bodies. Molluscum virus. Right? Write down owl's eye appearance. Owl's eye appearance is seen in cytomegalovirus. Write down cytomegalovirus. Fine? Okay. Right? Write down. So this is all about inclusion bodies. This is all about inclusion bodies. So now we'll go to the laboratory diagnosis of virus. We'll go to the laboratory diagnosis of virus. So if a virus has come into my cell, right? If let's say a virus has come into my cell, how will I detect it? Why are you late? If a virus has come into my cell, how will I detect it? Either I can detect the virus itself, or I can detect the virus proteins. Virus will release some proteins, no? I detect those virus proteins, correct? Or else I will detect the virus antibodies. If for these proteins there will be antibodies produced, no? Correct? If this is an antigen, if this is a problem, kuch to cure hoga, there will be some cure. So we will detect that cure, fine. Or we can detect the cytopathic effect, like the owl's eye appearance in kidney, that we can detect. So cytopathic effect we can detect, right? Or we can detect the DNA or RNA of the virus. DNA or RNA. When we were all infected with COVID virus, what is COVID? COVID was caused by what virus? No, it's a communicable disease caused by which virus? What is the coronavirus. Coronavirus is a SARS-CoV-2 type of virus. It is a SARS-CoV-2 type of virus. It is the classification. Fine. It comes from a larger classification called as MERS virus, right? Middle Eastern Respiratory Syndrome, right? It comes from there. So, this was a communicable disease. What did they do when they were testing for you for this virus? What are the tests they did? No swab. For what? RT-PCR. Correct? So, RT-PCR was done to detect what?
For identifying
No, that is correct. But what exactly were we detecting in that? We were trying to detect the DNA or RNA. Fine? Right? Okay. At the same time, you can detect the proteins, like if it's enzymes, if it has enzymes, right? So we can check those enzymes. So these are the whole processes, fine? Mainly we have a lot of processes like RT-PCR. At the same time, we can do ELISA. Fine? For the antibodies, we can do the ELISA. Remember ELISA?
Yes, sir.
We can do ELISA. At the same time, we can check for the, take a histopathologic sign, histopathologic slide, and then check for the cell cytopathic effect itself, like if it's a owl's eye appearance, that we can do, right? So, and there will be certain protein markers. If a virus is multiplying very much in our body, the body will respond to it with a lot of proteins released from the liver. So, from the liver, So, from the liver, there will be a lot of proteins released. Those proteins can be recognized. So, I'll tell you a list, just write it down. For direct demonstration, write down for virus. For detecting, write down detecting Then write down main heading virus. Detecting virus. We'll do electron microscopy. We'll do electron microscopy. Fine. Is a virus visible to the eye? Naked eye? No. Is it visible under a compound microscope? No. So we need a electron microscope. So we need electron microscope. Fine. Then write down write down immunoelectron microscope. Immunoelectron microscope. Immunoelectron microscope. Right? Write down fluorescent microscope. Fluorescent. Fluorescent microscope. The O should come here. Right? Fluorescent microscope. Fine. Now write down to detect the viral antigens. We're done with virus. Now we have to detect the antigens. Write down ELISA. Write down ELISA. Write down direct IF. Direct IF. Direct IF. Right? Write down now for specific antibodies, for virus antibodies. Write down the heading virus antibodies, and then write down again ELISA. In ELISA we can do both enzyme antigen detection and antibody detection, right? Remember sandwich technique, direct ELISA, indirect ELISA. So we can also do ELISA. Fine. Write down neutralization test. Neutralization test. Under this heading, write down neutralization test and ELISA. Right? Right? Now, write down to detect the genome. To detect the genome. To detect the genome. Right? Write down RT-PCR. RT-PCR. Real-time RT-PCR. What is PCR? Chain reaction. Write down. Reverse transcriptase PCR. Reverse transcriptase PCR. There are some viruses which you know what they do is once they go into the cell, they'll convert their RNA into DNA. They'll convert their genome is RNA, but they'll create a DNA. From that DNA, they'll again produce an RNA. So, to produce an RNA to DNA, you need a reverse transcriptase. To convert RNA to DNA, you need a reverse transcriptase. So, write down reverse transcriptase, PCR. Write down nucleic acid probe. Nucleic acid probe. Nucleic acid probe. P-R-O-B-E probe. Right? Okay. And lastly, you can do isolate the virus. Sorry. Write down lastly, isolate the virus by egg inoculation. Egg inoculation. Isolate the virus by egg inoculation. Write down in bracket embryonated. Write down embryonated. Fine. What happened? Right. Wrote down? Artha aita? So I'll barsiradella artha aita? Did you people understand everything? In nucleic acid probe, what they do is they take a micro needle. They go into the nucleic acid and then they collect sample and then they take the probe. Fine, that is one thing. In embryonic egg culture, what they do is they take any organism's egg, small egg, which is fertilized. A sperm and an ovum is necessary to fuse and produce a zygote. That will produce an embryo. Correct? In that embryo, they will inject the virus. They will inject the virus that will grow there. That virus will grow there. Some viruses grow in the placenta. Some viruses just grow in the head part, like the growing embryo. Like let's say there is a small embryo like this. Something like this. Right? Some viruses will just be very specific to this place, to the placenta. Some to the top part, some to the bottom part. Based on that, they tell which virus it is. They'll tell which virus it is, right? So that is embryonic egg culture. Sometimes what we do is we take a sheep blood. We take one RBC and then we inject a virus. Then we check for the growth and then decide which virus it is. Fine? Okay, that's all about lab diagnosis. Fine? Okay. Okay, write down another small thing called as continuous cell lines, CCL. I'll tell you what it is first. See, what are continuous cell lines is, sometimes we have to grow the virus. We know what are the properties of virus? What are the properties of virus we know? Can they grow in the free environment? They need a host. Now, when you are culturing a virus in the lab, like in big laboratories, when you are culturing the virus, what we try to do is, we need like unlimited supply of cells. We need more cells, right? So that we have to culture a virus. Like during COVID times, we would have needed so many cells, right? To culture the virus. Because at every part of the virus, we need. See, if the virus was staying outside, we can put dye and all and check. But this time virus has to be within a cell. It has to keep growing. So that we understand, okay, what are the proteins it produces, what are the DNA type of multiplication it does. That's why we create a cell line called as continuous cell line. We create something called as, we take cancerous cells. We take specific cancerous cells, and cancerous cells have a specific property called as automatic replication. They have something called as, all you have to do is just give them food. They'll keep growing. That is the best thing about cancerous cells. So now what we do is, we take cancerous cells and we inject virus. Cells will keep growing, virus will keep multiplying. So we have unlimited sources of cancerous cells. In that cells, these viruses are growing. Understood? See, simple point is that ನಮಗೆ ಕಲ್ಚರ್ ಯಾವತ್ತೂ ಕಮ್ಮಿ ಬೀಳಬಾರ್ದು. ಮತ್ತೆ ನಾವು ವೈರಸ್ ಎಲ್ಲ ಪ್ರಾಪರ್ಟೀಸ್ ತಿಳ್ಕೋಬೇಕು. ಅದಕ್ಕೋಸ್ಕರ ಏನು ಮಾಡ್ತೀವಿ, ಅನ್ಲಿಮಿಟೆಡ್ ಸಪ್ಲೈ ಆಫ್ ಸೆಲ್ಸ್ಗೋಸ್ಕರ ಕ್ಯಾನ್ಸರಸ್ ಟಿಶ್ಯೂ ತಗೋತೀವಿ. ಕ್ಯಾನ್ಸರಸ್ ಟಿಶ್ಯೂ ಆಟೋಮೆಟಿಕ್ ಆಗಿ ಮಲ್ಟಿಪ್ಲೈ ಆಗುತ್ತೆ. ಸೊ ಅದನ್ನು ಮಲ್ಟಿಪ್ಲೈ ಆಗಕ್ಕೆ ಬಿಟ್ಬಿಟ್ಟು ಎನರ್ಜಿ ರಿಸೋರ್ಸ್ ಕೊಟ್ಬಿಟ್ಟು ವಿಲ್ ಕೀಪ್ ಆನ್ ವೈರಸ್ ಆಟೋಮೆಟಿಕ್ ಆಗಿ ಮಲ್ಟಿಪ್ಲೈ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಸೊ ನಾವು ಆರಾಮಾಗಿ ಎಷ್ಟು ಬೇಕೋ ಅಷ್ಟು ಸೆಲ್ಸ್ ಅನ್ನು ತಗೊಂಡು ವೈರಸಸ್ನ ಸ್ಟಡಿ ಮಾಡ್ಬೋದು. ರೈಟ್? ಅಂಡರ್ಸ್ಟುಡ್ ವಾಟ್ ಇಸ್ what do you call it? Cell line, continuous cell line? Okay, I'll tell the definition, just write down. These are derived from cancerous cell lines. These are derived from cancerous cell lines. Fine? They have haploid chromosome. They have haploid chromosome. Fine? They are used to culture viruses. They are used to culture viruses. Examples, McCoy cell line. McCoy, McCoy, McCoy cell line. Hella cell line. Hella cell line. McCoy, McCoy, McCoy cell line, right now. Hella cell line. HELA, Hella. Just write down another thing. Write down another heading, cytopathic effect. Cytopathic, you understood what is cytopathic effect? ಅವಾಗಲೇ ಹೇಳಿದ್ದಲ್ಲ, what is that? ಹೇಳು. Cytopathic effect ಅಂದ್ರೆ inclusion bodies produce ಮಾಡುತ್ತೆ, that process is called as cytopathic effect. Write down cytopathic effect heading, right? First, degeneration. Degeneration happens in enterovirus. Write down degeneration enterovirus. ಅಂದ್ರೆ ಎಂಟೆರೋವೈರಸ್ ಕಲ್ಚರ್ ಮಾಡಿದಾಗ ಅವು ಸೆಲ್ಸ್ ಗಳನ್ನ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡುತ್ತೆ, ಕರೆಕ್ಟ್? ಓಕೆ. ಅದೇ ಏನಾದರೂ ನೀವು ಹರ್ಪಿಸ್ ಸಿಂಪ್ಲೆಕ್ಸ್ ವೈರಸ್ ತಗೊಂಡ್ರೆ, ಇಫ್ ಯು ಟೇಕ್ HSV, ಯು ಟೇಕ್ ಅ ಸ್ಲೈಡ್ ಅಂಡ್ ಲುಕ್ ಅಟ್ ಇಟ್, ಯು ವಿಲ್ ಸಿ balloons, you will see ballooning, right? Okay. Next, write down HSV, herpes simplex virus. ಏನಾಗುತ್ತೆ? What is the cytopathic effect? Ballooning. Write down ballooning. Write down RSV, respiratory syncytial virus. RSV cause, write down hyphen syncytium. Syncytium. Syncytium ಅಂದ್ರೆ ಏನು ಗೊತ್ತಾ? Listen, all eyes on good. Let's say these are multiple cells, 1 cell, 2 cell, 3 cell, 4 cell, 5 cell, and these dots are nucleus. These dots are prati cell ಗೆ ಒಂದು nucleus ಇರಬೇಕು. So this cell has a nucleus. There are how many nucleus? 5 nucleus. In a syncytium, now how many cells are there? Only 1 cell, but how many nucleus are there? 5. This is called as a syncytium. This process is called as a, or this structure is called as a syncytium, right? So in respiratory syncytial virus, it causes syncytium. It causes, right? Okay. Okay. So we are done with the part of. So what and all did we learn today? Inclusion bodies, cytopathic effect, laboratory diagnosis, correct? And we also spoke about cytopathic effect and inclusion bodies. Now we'll talk about the treatment. See, any virus, if it infects us, it will multiply. So how will you stop a virus defect? Like let's say you have got common cold, which is caused by rhinovirus. What do you do for rhinovirus? ಯಾರಿಗೂ ಕಾಮನ್ ಕೋಲ್ಡ್ ಬಂದಿಲ್ಲ, ಸ್ಟೀಮ್ ತಗೋತೀರಾ, ಮತ್ತೆ ವೈರಸ್ ಎಲ್ಲಿ ಸಾಯಿಸ್ತಾ ಇದ್ದೀರಾ? ವೇರ್ ಆರ್ ಯು ಕಿಲ್ಲಿಂಗ್ ದ ವೈರಸ್? ಯೂಶಲಿ the common cold, that is rhinovirus infection, does not have any treatment. It does not have any treatment. 7 days automatically it will decrease. In 7 days your common cold will automatically come down. Our immunity will fight it. But let's say you have got herpes infection. You have got, how does herpes infection happen? Unprotected sex. What is the cause? Unprotected sex. So let's say you have herpes infection, the herpes virus will be dividing, correct? It will be dividing. Now to stop the multiplication of the virus, what will you do? You have to decrease the whole process. You know what is the, remember what is the virus multiplication process? A cup ASMR. Remember A cup ASMR? What is A? Absorption. P, penetration. U, uncoating. B, biosynthesis. AS, assembly. R for release. So in this process, if we cut down any step, if we stop any step, the virus will stop replicating. So the modality of treatment is, if you give any drug, it has to stop this process. Most of the drugs stop the enzymes. They stop the enzymes. Like there is a DNA. Now this DNA has to multiply, but it needs an enzyme like DNA polymerase. If it is an RNA, it should need a RNA polymerase. Correct? So these drugs will act upon DNA polymerase or RNA polymerase. Most of the drugs. I'll tell the examples, just write down. Cidofovir, Cido, C-I-D-O, Cidofovir, Cidofovir, C-I-D-O-F-O-V-I-R, Cidofovir, Acyclovir, Acyclovir, Foscarnet, Foscarnet, F-O-S-C-A-R-N-E-T, Foscarnet, Acyclovir aita, Foscarnet aita, and what is the first thing I told? Write down. Right? Okay. Write down. Ribavirin, R-I-B-A-V-I-R-I-N, Ribavirin. Fine. Write down. Zanamivir. Sorry. Yeah, Zanamivir. It's Z-A. You can easily remember. Zanamivir. We made it Zanamivir. Z-A-N-A-M-I-V-I-R. Usually antiviral drugs ಎಲ್ಲಾದ್ರೂ suffix vir ಇರುತ್ತೆ. ಏನಿರುತ್ತೆ? Vir. So that it will represent this thing. So what are the drugs we know today? Cidofovir, Acyclovir, Foscarnet, listen, see, stopping virus in this whole APAB ASMR, you can stop any process. In this, but I have told you some drugs will act upon DNA polymerase, some drugs will act upon different processes. That is again a big topic, like which place it will act, what exactly will happen. So just remember this. Fine? Okay. Fine? No treatment. That is the treatment. Those are the examples. Those are the drugs. Right? If somebody has got a herpes infection, you give them ACE. Fine? Okay. If they are very close to you, give them Zanamivir. Okay. So what are all topics that we do today? Okay, we'll talk about another very important topic and then we'll end the class. So we'll talk about bacteriophages. We'll talk about bacteriophages. Fine. So what are bacteriophages, as I told you? The virus that attack the bacteria. Now you will ask me, sir, what is the difference between a virus attacking my cell and attacking a bacteria? Nam cell na yav tara attack maduತ್ತೋ ಅದೇ ತರ normal cell na attack ಮಾಡಬೇಕಲ್ಲ, bacterial cell na attack ಮಾಡಬಹುದು. Right? But there is one difference. There is something called as lytic cycle. There is something called as lysogenic cycle. There is something called as last bench. A virus, when it attacks human cell, it usually undergoes the lytic cycle. Human cell, most probably what happens is lytic cycle. But in bacteria, something called as lysogenic cycle happens. What happens? Lysogenic cycle. Okay. So what are the steps of multiplication of a virus? What are they? Absorption. Penetration. Up. We'll try to kind of decipher it here. A bacterial cell, fine. Our virus has come, right? Okay. Tell the first step. Absorb, right? Next. Penetration. So fine. Penetration. What goes in during the process of penetration? Nucleic acid plus capsomeres. Fine. Okay, this has gone in. Then what happens? Uncoating. So okay, the nucleic acid is free now. Then biosynthesis. That is by using machinery to produce DNA and RNA. Fine. Now here the change comes. Here the catch comes. In human beings, this biosynthesis happens. It produces again multiple viruses. Like let's say the star is a virus. Let's say it produces multiple viruses. Then lastly it kills this cell. It usually kills this cell and produces multiple viruses. Fine. These viruses will again go attack another cell. It will multiply, it kills this cell, done. It goes to another cell, infects. Right? But in bacteria, what does this? That is the lytic cycle. Lysis andre breakdown. Lysis andre? So here it breaks down, the cell goes. That is lytic. In lysogenic, what it does is, it is very smart. What is it? Very smart. Like all these first bench people and the last bench people. In middle people are a lot smarter. Only the first bench people and the last bench people are smart. They are very smart because they listen to the class. Last bench people because they escape the class. Fine. So what happens is, okay, listen. What it does is, it will integrate itself. The virus will integrate itself with the bacterial virus, bacterial genome. Let's say this is my Let's say this is the bacterial genome. Circular. Okay, listen. Let's say this is the bacterial genome. This is the bacterial genome. Let's say this is the virus genome. In lysogenic cycle, what it does is, this DNA will come and bind with it. It will come and it will become its own DNA. So bacterial DNA, this is viral DNA. It will become a fusion. First pure very bacterial DNA. This is virus DNA. Now what virus DNA does is, through this bacterial DNA it will come and bind up. Now this is the new fusion DNA. This is the new fusion DNA multiply ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಈ fusion DNA so multiple fusion DNAs are produced. So sudden ಆಗಿ in fusion ಆಗಿರೋ DNA ತನಗೆ ಇಷ್ಟ ಬಂದಿದ್ದ ಟೈಮಲ್ಲಿ it will break down. Whenever it wants, it will break down and it will start autonomously again producing. Understood? So lytic cycle is primary, but lysogenic cycle is secondary. But again it can come back to lytic cycle. ಇಷ್ಟ ಬಂದಾಗ ಫ್ಯೂಸ್ ಆಗುತ್ತೆ, ಬೇಡದೆ ಹೋದಾಗ ಬಿಟ್ಕೊಂಡು ಆರಾಮಾಗಿ ತಿರ್ಗಾ ಸೆಲ್ ನ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡುತ್ತೆ, ಇನ್ನೊಂದು ಸೆಲ್ ನ ಅಟ್ಯಾಕ್ ಮಾಡುತ್ತೆ. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? So that is the difference between human cell and bacterial cell. In us, mostly lytic cycle happens. In bacteria, lytic and lysogenic, both cycles happen. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? Now, sir, ಕೇಳಬಹುದು ಸರ್, lytic cycle, lysogenic cycle uses ಏನೇನು? In lytic cycle, it destroy the cell. That is okay, it's fine. काम हो गया तो destroy कर दो, हो गया. Lysogenic cycle, what is the benefit, sir? See, what it does is, sometimes, these normal antibacterial drugs like penicillins, cephalosporins, right? Amoxicillin. So what happens is, some bacteria, this pen is working better, I would have shown you better. I can't draw well. No, this is a very genuine problem. No, I'm not blaming him. All I'm saying is just try to make sure you get the right pen. Decreases my creativity. Okay, let's listen. So this is the bacterial genome. Let's say there is one part of it which has resistance. So ನೀನು ಎಷ್ಟೇ ಪೆನ್ಸಿಲಿನ್ ಹಾಕಿದ್ರು ಇದು ಈ ಪಾರ್ಟ್ ಅದನ್ನ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡಿಬಿಟ್ಟು ಪೆನ್ಸಿಲಿನ್ ಇಂದ ರೆಸಿಸ್ಟೆನ್ಸ್ ಮಾಡ್ತಿರುತ್ತೆ ಬ್ಯಾಕ್ಟೀರಿಯಾಗೆ. ಈಗ ವೈರಸ್ ಏನ್ ಮಾಡ್ತು, ತಂದೆ ಆದಂತ ಈ ಜಾಗ ಮಾಡ್ಕೋತು. ಇಟ್ ಗಾಟ್ ಫ್ಯೂಷನ್. ನೋ ಲೈಸೋಜೆನಿಕ್ ಸೈಕಲ್ ಅಲ್ಲಿ ಫ್ಯೂಷನ್ ಆಯ್ತು ತಾನೆ. ನೋ ವಾಟ್ ದಿಸ್ ವೈರಸ್ ವಿಲ್ ಡೂ ಇಸ್, ಇಟ್ ವಿಲ್ ಟೇಕ್, ತಾನು ತಿರ್ಗಾ ಸೆಪರೇಟ್ ಆಗ್ತಿರ್ಬೇಕಾದ್ರೆ ಇದನ್ನು ಕಿತ್ಕೊಂಡು ಹೋಗುತ್ತೆ. ಈ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನು ಸೆಪರೇಟ್ ಆಗಿ ತಗೊಂಡೋಗುತ್ತೆ. So that virus will have antibacterial or drug resistance property. ಅರ್ಥ ಆಗ್ತಿದೆಯಾ? See, this is a bacterial DNA. ಅದಕ್ಕೆ ರೆಸಿಸ್ಟೆನ್ಸ್ ಇದೆ. ಪೆನ್ಸಿಲಿನ್ ಹಾಕಿದ್ರೆ ಸಾಯಲ್ಲ. ಯಾಕೆ? ಅದಕ್ಕೆ ರೆಸಿಸ್ಟೆನ್ಸ್ ಇದೆ. ಈಗ ಏನ್ ಮಾಡುತ್ತೆ ವೈರಸ್, ಇಂಟೆಲಿಜೆಂಟ್ ಆಗಿ ಬಂದು ಅಟ್ಯಾಚ್ ಆಗುತ್ತಿದೆ. ಕರೆಕ್ಟ್? Now while going back, it will take this genome also. ಸೊ ಅಲ್ಟಿಮೇಟ್ಲಿ ಏನಾಗುತ್ತೆ, ವೈರಸ್ಗೆ ಇವಾಗ ನೀವು ಪೆನ್ಸಿಲಿನ್ ಹಾಕಿದ್ರೆ ಡೆತ್ ಆಗಲ್ಲ, ಬಿಕಾಸ್ ಇಟ್ ಟುಕ್ ದ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್. ಈ ತರ ಈ ವೈರಸ್ ತನಗೆ ತಗೊಳ್ಳೋದು ಅಷ್ಟೇ ಅಲ್ಲದೆ ಬೇರೆ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಸ್ಪ್ರೆಡ್ ಮಾಡುತ್ತೆ. ಈಗ ನೋಡಪ್ಪ, ಇದು ಯಾವುದು ಪೆನ್ಸಿಲಿನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಇದ್ದಿದ್ದು ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಇಲ್ಲೊಂದು ನಾರ್ಮಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದೆ, ಇದು ನೀವು ಪೆನ್ಸಿಲಿನ್ ಹಾಕಿದ್ರೆ ಸತ್ತೋಗ್ತಿದೆ. ವಾಟ್ ಇಸ್ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ವಿಲ್ ಡೂ ಇಸ್, ಇಟ್ ವಿಲ್ ಅಟ್ಯಾಕ್ ದಿಸ್ virus, it will take this gene, now it will attack this this virus, this bacteria. This bacteria will get penicillin resistance. This is how all the bacteria will start getting resistance. ಅರ್ಥ ಆಗ್ತಿದ್ಯಾ? Understood? ಯಾರ್ಯಾರು ಅರ್ಥ ಆಗ್ತಿರೋರು? You can raise your hands. I have no problem. I can explain again. Right? That's my work. So, normal bacteria, resistance bacteria. Attach both of them, goes there, steals its resistance gene, goes and gives to normal bacteria. That will also have resistance. That is the use of lysogenetic cycle. That is the use of ಗೊತ್ತಾಗಿದ್ರೆ ಓನ್ ವರ್ಡ್ಸ್ ಅಲ್ಲಿ ಬರ್ಕೊಡಿ, ಸೊ ದಟ್ ನಾಳೆ ನೀವು ಓದ್ತಾ ಇರ್ಬೇಕಾದ್ರೆ ಗ್ಯಾಪ್ತಾ ಇರಬೇಕು. ವಿಡಿಯೋಸ್ ಅಲ್ಲಿ ಅಪ್ಲೋಡ್ ಯು ವಿಲ್ ವಾಚ್, ದಟ್ ಇಸ್ ಅ ಡಿಫರೆಂಟ್ ಸ್ಟೋರಿ, ಬಟ್ ಬರ್ಕೊಂಡ್ರೆ ಬೇಗ ರೀಕಾಲ್ ಆಗುತ್ತೆ. ಫೈನ್, ಓಕೆ. ಇನ್ನೊಂದಿದೆಲ್ಲ ಏನು ಡಿಫರೆನ್ಸ್ ಇಲ್ಲ, ಸೇಮ್ ಎ ಪ ಬಿ ಎಸ್ ಮಾರೇ ಬರೀಬೇಕು. ಅಟ್ಯಾಚ್ಮೆಂಟ್, ಪೆನಿಟ್ರೇಷನ್, ಅನ್ಕೋಟಿಂಗ್, ಬಯೋಸಿಂಥಸಿಸ್, ಸೋ ಆನ್ ಅಂಡ್ ಸೋ ಫೋರ್ತ್. ಫೈನ್. ಸೊ ಜಸ್ಟ್ ರೈಟ್ ಡೌನ್ ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಆಫ್ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜಸ್. ಒಂದು ಸ್ಮಾಲ್ ಹೆಡ್ಡಿಂಗ್ ಬರ್ಕೊಳ್ಳಿ, ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಆಫ್ ಯಾಕೆ ರೆಕಾರ್ಡ್ ಆಗ್ತಿಲ್ಲ. Significance of bacteriophages. First, write down phage typing. Write down phage typing. P-H-A-G-E. Phage typing. T-Y-P-I-N-G. Phage typing. Right? Second point. Write down phage therapy. Phage therapy. Right? Then write down cloning vector. Cloning vector. ಈ ಲಾಸ್ಟ್ ಪೇಜ್ ಅಲ್ಲಿ ಬರೆಯೋದು ಕಂಡ್ರೆ ನನಗೆ ಆಗಲ್ಲ. ಆ ಶೀಟ್ ಎಲ್ಲಿ ಹೋಗೋದು ಗೊತ್ತಿಲ್ಲ, ಪರಿಚಯ ಕೊಂಡ ಇಟ್ಕೊಳ್ಳೋದು ಗೊತ್ತಿಲ್ಲ. Write down cloning vector. C-L-O-N-I-N-G. Cloning vector. Next you have transfer of drug resistance. Transfer of drug resistance. Transfer of drug resistance. ಇದರಲ್ಲಿ ಯಾವ್ದಾದ್ರು ಪಾಯಿಂಟ್ ಅರ್ಥ ಆಗಬೇಕಿತ್ತು. ಫೇಜ್ ಟೈಪಿಂಗ್ ಅಂದ್ರೆ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ನ ತಗೊಂಡು ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನು ಅಂತ ಕಂಡುಹಿಡಿಬಹುದು. ಸೈಡ್ ಈಗ ನೋಡಪ್ಪ, ಲೈಕ್ ನನ್ನ ಹತ್ರ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಇದೆ, ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಎ ಅಂತ. ನನ್ನ ಹತ್ರ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಇದೆ, ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್ ಹೆಸರೇನು? ಅದು ಬರೀ ಸ್ಟ್ರೆಪ್ಟೋಕಾಕಸ್ಗೆ ಇನ್ಫೆಕ್ಟ್ ಮಾಡುತ್ತೆ. ಸೊ ನನ್ನ ಹತ್ರ ಮಿಕ್ಸ್ಡ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಇದೆ, ಸ್ಟ್ರೆಪ್ಟೋಕಾಕಸ್, ಸ್ಟಾಫ್ಲೋಕಾಕಸ್, ನಿಸೆರಾ ಗೊನಾರಿಯಾ. ನಾನು ಮೇಲಿಂದ ಫೇಜ್ ಬಿಡ್ತೀನಿ, ಫೇಜ್ ಎ ಬಿಡ್ತೀನಿ, ಅದು ಹೋಗಿ ಎಲ್ಲೆಂಟು ಕೊಡುತ್ತೆ? ಸ್ಟ್ರೆಪ್ಟೋಕಾಕಸ್ಗೆ. ಸೊ ನೀನು ಬೇಸಿಕ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಿದ್ಯಾ ತಾನೆ? ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಫೇಜ್ ಟೈಪಿಂಗ್. ರೈಟ್? ಅದಾದ್ಮೇಲೆ ಫೇಜ್ ಟ್ರೀಟ್ಮೆಂಟ್. ಟ್ರೀಟ್ಮೆಂಟ್ ಆಫ್ ಥೆರಪಿ. ಈ ಬೇಸಿಕ್ ವರ್ಡ್ಸ್ ಅನ್ನ ಕ್ಯಾಚ್ ಮಾಡಿ. ಸೊ ಟ್ರೀಟ್ಮೆಂಟ್ ಅಲ್ಲಿ ಫೇಜ್ ಥೆರಪಿಲಿ ಏನು ಮಾಡ್ತೀವಿ ಅಂದರೆ, ಫೇಜ್ ಒಳಗಡೆ ವಿಲ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಅ ಸ್ಪೆಸಿಫಿಕ್ ಜೀನ್. ಆ ಜೀನ್ ಹೋಗ್ಬಿಟ್ಟು ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡುತ್ತೆ. ಅರ್ಥ ಆಗ್ತಿದೆಯಾ? ಲೆಟ್ ಸೇ ಐ ಹ್ಯಾವ್ ಟೇಕನ್ ಸ್ಟ್ರೆಪ್ಟೋಕಾಕಸ್, ಐ ಹ್ಯಾವ್ ಟೇಕನ್ ಅ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಐ ಹ್ಯಾವ್ ಟೇಕನ್ ಅ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್, ಐ ಹ್ಯಾವ್ ಪುಟ್ ಸಮ್ gene. This gene will go to the bacteria, infect the bacteria, and that gene will be introduced, and that total bacteria will be destroyed, like an atom bomb basically. Right? Like an atom bomb. Right? Cloning vector andre it's again a big topic. Again, same gene transfer ಆಗುತ್ತೆ. Exactly. If somebody has read 12th ಅಲ್ಲಿ there was something, 12th ಅಲ್ಲಿ PUC science ಅಲ್ಲಿ there was something called as RDNA, recombinant DNA technology. ಅದು ಓದಿದ್ರೆ ಅರ್ಥ ಆಗುತ್ತೆ ಕ್ಲೋನಿಂಗ್ ವೆಕ್ಟರ್, ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್. ಜ್ಞಾಪಕ ಇದೆಯಾ ಬ್ಯಾಕ್ಟೀರಿಯೋಫೇಜ್? ಹೌದಿಲ್ಲ, ಎಲ್ಲರೂ ಸೈನ್ಸ್ ಓದಿದ್ದಾರೆ. ಅಲ್ಲ 12th ಅಲ್ಲಿ ಕಾಮರ್ಸ್ ಪೀಪಲ್ have also come to BSc nursing. BSc nursing people science. Okay, right? So that's it guys. So ಇವತ್ತಿಗೆ I guess general virology is done. I guess general virology ನೇ ಬೇಕಾಗಿತ್ತು ನಿಮಗೆ. ಸ್ಪೆಸಿಫಿಕ್ ಆಗಿ ವೈರಸಸ್ ಬಗ್ಗೆ ಏನು ಚಾಪ್ಟರ್ ಇಲ್ಲ. ಸೊ ಲಾಸ್ಟ್ ಕ್ವಿಕ್ ಸಮರಿ ತಗೋಣ. ವೈರಾಲಜಿ ಬಗ್ಗೆ ಏನೇನು ಮಾತಾಡಿದ್ರೆ, ಸ್ಟಾರ್ಟಿಂಗ್ ಅಲ್ಲಿ ವಿ ಸ್ಟಾರ್ಟೆಡ್ ವಿತ್ ವೈರಸ್ ಪ್ರಾಪರ್ಟೀಸ್. ವೈರಸ್ ಅಲ್ಲಿ ಏನೇನು ಪ್ರಾಪರ್ಟೀಸ್? ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಫಿಲ್ಟರ್ಸ್ ನ ಪಾಸ್ ಆಗುತ್ತೆ, ತುಂಬಾ ಸ್ಮಾಲ್ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್, ಹೋಸ್ಟ್ ಸೆಲ್ ಬೇಕೇ ಬೇಕು ಡಿವೈಡ್ ಆಗೋದಕ್ಕೆ. ಆಫ್ ಮಾಡ್ಬಿಟ್ಟ? ಆಫ್ ಮಾಡಿದಲ್ಲ, ರೈಟ್? ಯಾಕಂದ್ರೆ ಲಾಸ್ಟ್ ಸಮರಿ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಸೊ ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅಥವಾ ಯಾವುದೋ ಹೋಸ್ಟ್ ಸೆಲ್ ಬೇಕೇ ಬೇಕು ಮಲ್ಟಿಪ್ಲೈ ಆಗಕ್ಕೆ ಅಂತ ಮಾತಾಡೋದು. ದೆನ್ ವಿ ಸ್ಕಿಪ್ಡ್ ಇನ್ ಟು ಟೈಪ್ಸ್ ಆಫ್ ವೈರಸಸ್. ವಿ ಸ್ಪೋಕ್ ಅಬೌಟ್ ಡಿಫ್ರೆಂಟ್ ವೈರಸಸ್, ಕ್ಲಾಸಿಫೈ ಮಾಡಿದ್ದೀವಿ ಬೇಸಿಕ್ಲಿ. ಎಂಟೆರೋ ವೈರಸ್, ಮಿಕ್ಸೋ ವೈರಸ್, ಹಾಗೆ ನಾರ್ಕೋಮಿಕ್ಸೋ ವೈರಸ್, ಪ್ಯಾರಾಮಿಕ್ಸೋ ವೈರಸ್, ರೆಸ್ಪಿರೇಟರಿ ಸಿನ್ಸಿಷಿಯಲ್ ವೈರಸ್, ಬೇಸಿಕ್ಲಿ ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಬರುತ್ತೆ. Then we want to understand how do viruses multiply. We spoke about how viruses multiply. It was APAB ASMR. Fine. Then what else did we speak about? We spoke about today's class where we spoke about we spoke about the symmetries of virus. We spoke about helical symmetry. We spoke about what is the icosahedral symmetry. We spoke about a complex symmetry. Fine. Then we spoke about the parts of a virus. Like we spoke about the outer. What is the outermost part?
Capsid.
No, not capsid. That girl who told me that especially part is that. What is the outermost covering? Envelope. Envelope ಅಲ್ಲಿ liquid ಎಲ್ಲಿಂದ ಬರುತ್ತೆ? From the host cell. Remember? Host cell proteins are from the viruses. Adadmelind we spoke about capsids, then we spoke about capsomeres, we spoke about the genetic material which could be DNA or RNA, we spoke about the multiplication. After the multiplication we spoke about inclusion bodies. These inclusion bodies process was called as cytopathic effect. We spoke about the cytopathic effect. Then we spoke about bacteriophages, and we also spoke about what are the drugs used to kill the viruses and try to stop the multiplication. So this overall is the overview of virology. This is the overview of virology. So whatever I told in class, try to note it down. How much ever you can, just write down. Like even while listening, write it down. So that like last exam time, if you skim, you easily get to know. Or else I'll keep uploading the videos. You can get it. Fine. Okay, it's done. Any doubts?
Like and
So today.